Any other questions? All right, let's work through the concept questions on this one. So, what is the first concept question? I wrote them, but I don't remember them.
Just read it out.
Yeah, read it out.
Okay. For many highly heritable traits, for example, height, children tend to have a trait value between that of their parents. For example, the child is shorter than the tall parent and taller than the short parent. Why is this?
Why is this?
Do you have a good answer?
I said because you inherit some variance from like the tall parent and some from the short parent, so it's kind of like a mix of both.
Exactly. Yeah, so your dad's taller, half of his taller variance you're gonna get, and if your mom's a little shorter, half of her slightly shorter variance you're gonna get, and so it averages out. And that works really nicely on a model like this, where your genetics don't interact with each other. And the reason height is one of the examples there is because height tends to actually be a trait that actually follows this model decently well. Not really, but like to a first order approximation, it has a lot of what we consider just these additive effects where the genetics don't interact. So yeah, half of whatever your genetics that are from your mom, half of whatever the genetics are from your dad, they come together, average out nicely, and there you go. So yes, there's a very good answer for that. All right, next question.
The next question is the one we just talked about, with like the random draw.
Okay, cool. Question after.
Should I read that now?
Yeah, I think good.
One of the most heritable traits is graduating from medical school. Is this inherited through genetics? If not, why does this trait run in families? Is that just environmental effects? Like you're more likely, like if it's, obviously it doesn't seem like it should be in your genome as to whether or not you graduate from med school, but you could have like a support system around you that's like maybe that's why you did it.
Exactly, like if your parents, or if one or both of your parents are doctors, you're gonna be like, what life do you want to go? I want to be like my parent. I'll be a
And
doctor.
you'll have money to go to med school.
And you'll have money to go to med school. So yes, there's a huge amount of environmental factors that are highly correlated with going to med school. And it just so happens that those environmental factors are very much dependent on your parents. And so this is a trait where it's super heritable, but not through your genetics. It's actually because your environment is correlated with your parents' environment, which is correlated with their parents' environment. And
I think your answer is still good though. Sorry, I'm reading over her shoulder.
Okay.
Because there's another consideration too, which is that there could be some element that is heritable, for example, intelligence. So you could be getting part of it from a genetic basis. Like we don't know.
that's true.
That one is pretty well confounded by environmental factors.
Yeah.
But there may still be an element of there being another phenotype that going to medical school is associated with.
Oh, I'm sure that there are genetics that are associated with this, but and so this is a really good thing for a module that's like a few away still. When I said that these things are heritable, I also earlier said that as computational people, we like to put numbers on things, and we actually have figured out how to quantify heritability. And so this is one of those traits where the heritability as a number is actually probably, or the genetic heritability is probably really, really small, which means, yeah, there's a little bit of genetics that plays a role, but it's like mainly environment. Whereas with something like height is like 80% heritable, which means 80% of your height is dictated basically by the genetics you inherit, and only 20% of it is from the environment. And so, yeah, so for something like med school, there probably is a few things like intelligence, and yeah, intelligence is something which like is slightly heritable.
That's a really difficult one to do genetics.
It's really hard to do.
Because there's not a good phenotype,
Yeah.
right?
We're really good with genetics for like the extreme versions, like if you have like autism or something like that. But for basically the general population,
Well, I mean, even autism genetics is pretty complicated.
but we know there's genetics.
Yeah.
But like if you just kind of look at like IQ scores, I think unless you really look at the tails, there's very little
Yeah.
genetics that can be like, oh, you're a 90 and you're like a 113. Like, we
These
might have
additive
something.
effects aren't a good representation of IQ scores, mostly because IQ scores is not very replicable, so it's
Yeah,
not a great measure.
yeah. So-
But just like quickly,
Uh-huh.
this isn't important, but like one of the ways that we can measure heritability is through twin studies. So you might have heard that, where we have two people who have the exact same environment, but if they're fraternal twins, then they have just sibling relatedness, and if they're identical twins, then they have identical genetics, and so you can look at how different they are in this phenotype, and that'll give you an idea of the heritability. So I'm sure Rob will talk a lot about other ways to measure heritability.
Yeah, when we get to here,
Yeah,
we'll go through all of this, but.
that's a good one to keep in mind.
Alright, cool. Next question.
Okay, assume there are M SNPs in the genome. If a true model for a trait is the additive one, so GB plus E, how many Bs are there?
Yeah. So if this is our true model, how many effect sizes are there?
M.
There's M, right? Because for every position or every genetic variant we look at, there's an M. Okay.
Another like interesting thing that relates to this is where do you get the betas from? So we'll talk about that more in another module, but usually you'll get them from a previous GWAS or something similar. Yeah.
Okay. And then the next question, final question.
Well, the next question is the same, but for the third model.
Exactly. So how many betas are there in this guy here? So let's count them up. So in the U, there's none, right? So what about this summation right here?
Yeah.
M, right? Because it's just one M for every, or one beta for every G you have. All right, here we have a gene by your diet interaction. So how many measures of your diet do you have for each person? Just one, right? Whatever this ED is. And so you're going to do whatever that is times your genotype for each genotype. And how many times are you going to do that? M times again. Right, so for this interaction, again, we only have M. So we have 1M, 2M. How about this environment interaction? It's just M again. Okay, and how about this gene by gene interaction? So M squared, right? So we're basically at M squared plus a couple linear M's that we don't care about. So this is going to be, yeah, essentially this is M squared, right? So if M is like, say, 1 million, which is not an unheard of M,
Wait.
how many betas are there?
10 to 12. Or some order around.
Yeah,
More
a
than
huge
that.
number,
Yeah.
right? So that's the real problem right here, is to actually look at all these interactions. We're doing like 10 to the 12 betas. And in statistics world, it's really hard to estimate lots and lots and lots of values. It's easy to estimate like one value. Say you sample 1,000 people and you need to estimate like one beta. It's like easy, right? You do some linear regression. But if you need to estimate 10 to the 12 betas, you just can't really do that. You don't have enough statistical power to do that. And so what this means is that it's really hard to estimate these values.
Especially if you add in
many.
another G, because then it's going to be 1 million to the third.
Yeah, and you can't,
Right, like-
if you do a G by G by G.
Because then
this
you
is
also
over.
have to add the 2 interaction plus the 3 interaction. So it's like
Yeah.
exponentially more. Not technically exponentially, but
Yeah, and so so this one's hard to do due to statistical limitations
but also like computational limitations.
and computational
Because it
limitations.
will take forever
Yeah.
and like use up a lot of memory.
Yeah, these ones are hard because just measuring environment tends to be hard. So basically, since these ones are all hard, we're left with this one easy thing, which is I say this is why this is the model we know and love, because it's statistically easy and it's easy to get the data for it. And so we just love to believe that this is the true model, even in reality, it is not the true
It's
model.
clearly not the
It's
true
clearly
model.
not the true model, But if you know how this model works, you basically can understand like 90% of the literature out there on genetics. And there's a lot of people now trying to understand where and how this model fails, because that's also something we don't understand. We know it's not good, but understanding why it's not good tends to actually be a more challenging problem than you would think it is as well. So if you come work with me, big summer, you get to work on this problem. All right, any other questions? I think that's it.
Wait, isn't there
There was
one more?
one more.
Oh, one more.
Yeah. Okay, I can read it again. Okay, assume you have two SNPs in perfect LD. One of them has a true effect of making people 1 inch taller, the other has no effect on height. You collect height measurements and the genotypes of the two SNPs for many individuals. How can you tell which SNP has the true non-zero causal effect and which no effect? Can you?
You cannot. Not from the data that you're collecting here. And that's because when there's perfect LD, the statistical tests you do on those two SNPs are going to give you identical results. Because it's the SNP that's changing, not your phenotype that's changing.
If this was something in like mice, you could try to knock one in or something, right?
Yeah, that's it. From the data we have.
Right.
If you actually think that one of these two is it, then what you do is you go talk to somebody who has like a wet lab, and they'll probably do like some cell lines, and they'll do like whatever magic they do, and they'll be like, it's this one.
Right.
But from the computational statistical side, two things in perfect LD, it's not impossible, but if we are able to say it's this one and not that one, it's only probabilistically that we can say that, and we're doing that using other data sources that we're pulling in. And it's more so we're guessing that it's one over the other based on some prior knowledge
Functional
we have.
information about what
Yeah.
it does.
Do you care though? Like if they're perfect LD?
You do if you want, oh sorry.
Yeah, go on.
You do if you want to like cure diseases or
But
like,
They're like perfect LD, so you should pick one, would not like.
right, so the reason we want to know what variants cause disease is because we want to be able to target them with like medicine or other things. So if you don't know which one is driving it, they might be in different genes or they might have different effects. So you don't know which one of those things to try and treat with medication or genetic stuff.
Yeah, LD is a statistical problem that it exists because of biology, but yeah, two SNPs in statistical LD with each other, yeah, biologically can have very, very different functions. And at the end of the day, we're using statistics to kind of guide us to the correct biology, and we're not great at that, but like, that's our goal.
You still need the biology to interpret the statistics,
Yeah.
basically.
So,
So you know what to do with that information. Because saying there's a variant here that's associated with disease does not tell you anything about the disease.
other than something near that variant somehow
Not
does
even near it
something.
though, like just something
Yes.
related to this variant might have an effect on
Yeah.
the disease.
So, all right. Anything else? All right. Thank you guys so much.